Our next presenter is Silas Jenkins. He's a graduate student in the biology department at Texas State University. The research presented today is for their master's thesis, which examines the role of mycorrhizae in oak seedling health. Si?
My thesis research is looking at if you were to provide an oak seedling with soil from different ecological site types, if that influences what mycorrhizal relationships they form, and how they tolerate water stress. So my questions, were basically, if you provide a seedling with soil from different situations, different habitat types, do they develop different partnerships or do they develop the same partnerships? And then on top of that, if you water stress them, does that influence which partnerships they form, and does that, or do some partnerships potentially benefit them more under water stress situations? So, and I look specifically at their growth and their photosynthetic rate. Before I go too much into my hypotheses, I want to address what assumptions informed this research, which one is actually somewhat contradictory to what Brian just said, was that I was operating under the assumption that oaks are dual mycorrhizal. There's, mixed stuff in the literature. It's hard to kind of parse, but there are some studies that show that oaks can form and benefit from arbuscular mycorrhizae or ectomycorrhizae, and that since arbuscular mycorrhizae are more ubiquitous and tend to be more generalist or in who they associate with, it's hard to generalize about, obviously, mycorrhizae, but arbuscular mycorrhizae are compatible with more species. So, and there are some studies that have shown that under drought scenarios, the same oak trees will shift in their relationships away from ectos towards arbuscular mycorrhizae. So I was operating under the assumption that they're dual mycorrhizal. I also had the idea that there could be overlap in the symbionts of juniper and oak. Since Texas red oak co-occur with juniper throughout most of their range, I thought they probably share some of the same arbuscular mycorrhizae. And also, I was assuming that mutualists would be limited away from a host plant. There are studies that show that as you move away from the forest edge and from ectomycorrhizal hosts, you're not going to see as high of a diversity or abundance of ectomycorrhizae. So I hypothesized that when provided with what I'm calling the home site soils, so this is from a mixed juniper oak forest, that the seedlings would do better in terms of growth and photosynthesis, and that basically the closer that inoculation treatment was to their home soil, you'd have more colonization, more diversity. But I also thought that if they were inoculated with juniper, soil coming from beneath a juniper in their home terrain, that they might have more of those drought-tolerant mycorrhizae, and they themselves might be more drought-tolerant. So to test these hypotheses, I conducted a factorial split-block greenhouse experiment with 4 different inoculation treatments, 2 different water treatments, and so each of those combinations was replicated 52 times. I started off by collecting acorns at Riker on the BCP and weighing them, and waiting until spring and selecting seedlings of similar size to participate in the study in the spring. So in the spring, when we transplanted them from these containers into liter pots, we added the Inocula. The Inocula came from 2 different tracks on the BCP. One is Riker Ranch, which I'm calling their home. That's where the acorns are from. And this map shows the blue shapes are the location of the oak trees that provided inoculum, and the red shapes are the location of the juniper trees. And so there were 16 to 20 trees. At each tree, we collected inocula from 2 different spots because aspect, like Brian was saying, they're patchily distributed, and things like aspect make a difference too. But basically, we're collecting from the base of the trees. These were elder trees, like basal circumference about 100 centimeters. This gives you a sense of their size. And so I forgot to say, so in the oak zone, it's very much mixed juniper oak canopy. In the juniper area, we were selecting from trees that were at least 15 meters from adult red oak. So I wanted to have a treatment that was not as entangled with oak roots. So the other site, which I'm calling the away site, is from Cortana, also on the BCP. And here we collected soil from what I'm calling the mott treatment. So Cortana was cleared in about 20 years ago of most woody vegetation. So these remnant motts that have juniper and Quercus fusiformis and clearings. So here I was collecting from underneath the motts, kind of crawling in there, and so within a short distance of both an oak, but a Quercus fusiformis and a juniper. And then in the clearings, we were several meters from the nearest ectomycorrhizal plant. So the seedlings were randomly assigned to a block and a position in a block, and half of the block was watered and half was droughted. But we let them establish for 7 weeks to make sure they formed connections with mycorrhizae before imposing the drought stress. And from there, the well-watered group was watered to field capacity once a week, and the droughted group was not watered at all for the duration of the study. And a little ways into the drought, I started collecting photosynthetic readings using that contraption. So it took 3 to 4 weeks before the well-watered and the droughted group separated in terms of their photosynthetic rate. These are surprisingly resilient little, it was really impressive. So about 3 to 4 weeks in, they separate. But there weren't a lot of inoculation treatment differences in terms of their photosynthetic rate. The blue arrow shows the one time where there were inoculation treatment differences, and that the graph on the right, maybe you can tell, but the juniper, the acronyms are clearing, C, J, juniper, M, mott, and O, oak. The juniper inoculated seedlings were photosynthesizing at a significantly higher rate than the mott and oak inoculated seedlings. So about sometime after, more than a month into the drought, they started showing like obvious visual signs of drought stress. And we were documenting that, their crispiness, their time to crispiness. And so this graph shows how they kind of diverged after a point, and the statistical test we ran indicates that there were significant differences. You can see that the red line is the juniper inoculated seedlings, the green is mott, the yellow is oak, and the purple is clearing. And the biggest difference obviously was between the juniper and the clearing. The juniper inoculated seedlings took longer to get crispy. Of course, after 2 months of being in a 1-liter pot in a greenhouse with a broken evaporative cooler, like everybody was crispy at that point, but there were differences in how that progressed throughout the course of the drought. So we harvested them and separated above ground and below ground biomass, and separated leaves from stems, and dried them and weighed them. And these graphs are showing that as far as leaf mass, there were not obviously the well-watered seedlings got bigger, but among the inoculation treatments, there weren't statistically significant size differences in leaf mass. There were in stem mass. The most significant difference was between the clearing inoculated seedlings, which had the biggest stem mass, and the mott inoculated seedlings had the smallest stem mass in the well-watered group. The droughted seedlings were all basically the same size. There's a marginally significant difference between the clearing inoculated and the oak inoculated, and the juniper were closer to the clearing. And The frozen roots got shipped on dry ice to Flagstaff, Northern Arizona University, and they graciously trained me in how to do the mycorrhizal microscopy. So I had, subsampled the root systems, different parts of the root systems, and then looking at a subset of that on a dish with a grid. And you're tracing the grid lines, and every time there's a intersection, you're counting whether it's living in mycorrhizal, dead in mycorrhizal, dead and non-mycorrhizal, dead and all those combinations. And then that's the colonization count. And it took me a while to figure out what's actually colonized. You can see the ectomycorrhizae under a light microscope. And then also I was tracking what the morphotypes, so based on their color, texture, shape, trying to, as a proxy for who they are. And I was tracking like more than 20 different groups, and it got a little overwhelming. But our buscular mycorrhizae, there's a different process of looking at them because you can't just see them under a light microscope. You have to clear and stain the roots. And I was surprised because I thought oaks would be dual, and I thought particularly under a high stress situation they might have AM. There weren't any. There were dark septate endophytes and other endophytes. Like you can see the dark septate endophytes. We could not see any. This is what you might see. You would see arbuscules maybe and vesicles and hyphae, but however, they were highly colonized by ectomycorrhizae regardless of the inoculation treatment. So I was impressed by that, that even out in the clearing, kind of far from an ecto host, that they still had a lot. We just got DNA sequences back on Sunday, so I'm kind of parsing how did my categories match up with what the DNA is saying, and that's all a little complicated. But for now, I can say that the ectos are primarily coming from 3 different families in Ascomycota. There's a couple of tubers. These are the closest, it may be a different species that actually hasn't been described yet that's closely related to these two, but there's a lot of tubers. There's a lot of Pyronimataceae family and some Pizziza. And also some weirdos, like there's a lot of this fungus that causes dandruff in humans. It's also a plant endophyte, and I'm a little bewildered by the data right now, but for now, my tentative conclusions are that, Texas red oaks might not actually be dual mycorrhizal, or it might be that establishment period that we gave them, that they preferentially establish ectos, and I don't know. And that the ectomycorrhizae, or at least these taxa, are pretty abundant and available even away from ectohosts and in, so that was interesting. It might be too that the kinds of mycorrhizae that can tolerate being in a tiny pot in a hot greenhouse away from a tree, away from their network, are more tolerant of different site types. And then to the extent that there were inoculation treatment differences, I still don't know whether to attribute that to the mycorrhizal, if there are mycorrhizal community differences. I think we'll get more clarity on that as I figure out the DNA stuff, or differences in the endophytes, or differences in pathogens too. So, but it does seem like, the couple of times that there were significant differences, the Juniper were photosynthesizing a little better at a pretty severe moment in the drought, and then they also didn't senesce quite as rapidly. So that was interesting to see. Thanks. This wouldn't be possible without a bunch of mutualisms that I participate in with my family, with my advisor, Susan Schwinning, and actually a lot of people who are in this room who, have guided me through this project, especially Lisa O'Donnell and Kitty Gehring's lab at NAU, and Lisa Markovchik and a bunch of other folks. So. That's a good question. I have all those numbers because the photosynthetic machine records it, and I don't totally remember. I was taking the readings early in the morning, because they actually just stopped photosynthesizing at some point. But it was hot. It made outside feel cool. So however hot that was, that's how hot it was. And it was pretty, yeah, I don't know. I wish I could tell you actual numbers, but yeah. I can say that the time to crispiness was less for the Juniper inoculated seedlings. And yeah, I just got this new deluge of information on Sunday, so I'm still like unsure. Based on my morphotyping, it seemed like, they're all highly colonized, and it seemed like they're colonized by similar mycorrhizae. So I was thinking that it might be a combination of particular endophytes that are more in the soil around Juniper, or maybe removal from pathogens, because the Juniper treatment was further from oaks, compared to the Mott and the oak treatment. So maybe a combo of, I don't know. If anyone has any insights about what they think it might mean, I'm soliciting input. So yeah. Oh, good question. Sorry, I realized I forgot to repeat the questions. The question was, how much material was I using to inoculate each pot? It was half a cup of soil and root fragments in a liter pot, and trying to like kind of pour it in so it was getting up and down the root column, if that makes sense. Yeah. Basically, yeah.